أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم فسيكفيكهم الله وهو السميع العليم صدق الله العظيم. Measurement of protein content of samples is an integral part of many medical research. Today I'm going to explain the most common current spectrophotometric methods for measurement of protein concentration of samples, followed by measurement of protein contents of Wistar rat liver, lungs, and kidneys by two famous widely used methods, Lowry and Bradford methods. I classified spectrophotometric protein measurement methods into two groups: direct spectrophotometric method and indirect spectrophotometric methods, also called colorimetric methods. In direct spectrophotometric method, ultraviolet light absorption of a pure protein sample is measured at 280 nanometer. The absorbance at this wavelength is directly proportional to protein content of the sample. In indirect spectrophotometric methods, a reagent commonly colored reacts with protein in the sample, and a colored product is formed whose visible light absorption is measured at appropriate wavelength. The intensity of visible light absorption is proportional to protein content of the sample. In both methods, appropriate protein calibration or standard curve should be constructed by the same method. The protein concentration of the sample is determined by interpolation of UV or visible light absorption at suitable wavelength over protein tabulated concentrations on protein standard curve. The first and the most rapid and simple but most likely inaccurate method is on the basis of ultraviolet absorption of a protein sample at 280 nanometer. Proteins are polymers of amino acids. Most amino acids do not absorb UV light. However, aromatic amino acids including tryptophan, tyrosine, and to a much lesser extent phenylalanine absorb UV light within 250 nanometer to 290 nanometer. This accounts for the characteristic strong absorption of UV light by most proteins at a wavelength of 280 nanometer, a physical property employed by researchers in the characterization of the proteins. Please pay attention to the slide. In this figure, you see ultraviolet light absorption spectra of aromatic amino acids tryptophan, tyrosine, and phenylalanine with equimolar concentrations of 1 millimolar at pH 6 and under identical conditions. The measured absorbance of tryptophan is more than 4 times that of tyrosine and more than 20 times that of phenylalanine at 280 nanometer. Note that the maximum light absorption for both tryptophan and tyrosine occurs near 280 nanometer. Ultraviolet light absorption by phenylalanine generally contributes little to the spectroscopic properties of proteins. Direct determination of protein concentration of a pure protein sample with a known molar attenuation coefficient is possible by measurement of the sample UV absorbance at 280 nanometer. The molar attenuation coefficients for many proteins have been determined so far. Because of the different mole fractions of tryptophan and tyrosine residues in different proteins, the molar attenuation coefficients of proteins vary significantly. The most common and important interfering compounds in this method are nucleic acids. Please pay attention that nucleic acids, both DNA and RNA, absorb UV light at 260 nanometer. Nucleic acids are polymers of nucleotides. Nucleotides consist of a sugar molecule, either ribose in RNA or deoxyribose in DNA, attached to a phosphate group and a nitrogen-containing base. Nitrogen-containing bases, including adenine, guanine, cytosine, and thymine in DNA and uracil in RNA account for UV absorption of nucleic acids at 260 nanometer. Please pay attention to the figure. In this figure, you see the UV spectra of adenosine monophosphate or AMP represented by blue curve, guanosine monophosphate or GMP represented by green curve, uridine monophosphate or UMP represented by orange curve, deoxythymidine monophosphate or DTMP represented by violet curve, and cytidine monophosphate or CMP represented by red curve. Please pay attention that purinic nucleotides are stronger UV absorbers than pyrimidinic nucleotides at 260 nanometer due to having an additional ring. In this figure, you see the spectra of pure protein sample represented by blue curve, pure nucleic acid sample represented by green curve, and a protein-contaminated nucleic acid sample represented by dashed red curve. Please take into account that the ratio of UV absorbance of 260 nanometer to 280 nanometer is commonly used to assess DNA contamination of protein samples. The reverse, however, is not true, as large amount of protein contamination is necessary to significantly affect the 260 nanometer to 280 nanometer absorbance ratio in a nucleic acid solution. Question: why? Please pay attention to the tables. As you see, protein does not change the 260 nanometer to 280 nanometer absorbance ratio of a nucleic acid sample significantly, but nucleic acids change this ratio of a protein sample significantly. Finally, I should state that contamination of protein sample by many substances can significantly induce inaccuracy in measurement of protein concentration by this method. Thus, this method is most suitable for pure protein samples. The most common current indirect spectrophotometric methods, also called colorimetric methods, include Biret method, Lowry method, BCA or biceinquininic acid method, and Bradford method, of which the first three are copper-based. Biret method is a method for detecting the presence of peptide bonds. The reaction was first observed by German scientist von Ferdinand Rose in 1833, which independently rediscovered by Polish physiologist Gustav Piotrowski in 1857. Peptides containing 3 or more amino acid residues will form a deep purple-colored chelate complex with cupric ions in an alkaline medium containing sodium-potassium tartrate. A similar colored chelate complex forms with the organic compound biuret and the cupric ion. Several variations of this test have been developed so far, such as the BCA and the Lowry methods. Biuret assay is the oldest and the least sensitive copper-based assay for measurement of total protein among colorimetric methods. Despite this advantage of the least sensitivity, Biuret assay is still frequently used due to advantage over other copper-based assays, including easy performance, easy preparation of reagents, and markedly less susceptibility to chemical interference. In general, Biuret assay is useful for samples with high protein contents between 1 to 10 milligrams protein per each milliliter of sample. Most proteins produce a deep purple color with this assay with maximum absorbance at 550 nanometer. Copper chelating agents and reducing substances interfere with this method. The Lowry method is another copper-based assay introduced by Oliver H. Lowry at 1951 based on the reaction of cuprous cation produced by the oxidation of peptide bonds with yellow Folin Siokalto reagent, a mixture of phosphotangostic acid and phosphomalipdic acid in the Folin Siokalto reaction to form a blue-colored reaction product whose absorbance is measured at 750 nanometer. Lowry assay nearly 100 times more sensitive than the Biret reaction alone. Chelating agents, surfactants, high potassium concentrations, reducing agents, and thiol-containing substances interfere with this method. The next method is BCA or biocinquenonic acid method. This method was introduced by Paul K. Smith and his colleagues in 1985. It is a modification of the Lowry method. The mechanism of color formation with proteins for the BCA reagent is similar to that of the Lowry reagent, but with several significant differences. In this method, cupric ions are reduced by peptide bonds to cuprous ions in an alkaline medium like Lowry method. Each cuprous ion thus formed is combined with two molecules of highly sensitive and selective cuprous cation chelator biocinquenonic acid or BCA to form a water-soluble purple-colored reaction product whose absorbance is measured at 562 nanometer. The primary advantage of the BCA protein assay reagent is that most surfactants, even if present in the sample at concentrations up to 5% volume by volume, do not interfere with the assay. Chelating agents, lipids or lipoproteins, reducing agents, thiol-containing substances, and biogenic amines or catecholamines interfere with this method. Bradford method is based on the reaction of protein molecules with Bradford reagent containing Kumasi brilliant blue G250 dye under acidic conditions to form a water-soluble blue-colored reaction product whose absorbance is measured at 590 nanometer. Bradford reagent has two absorbance maxima at 450 nanometer and 650 nanometer, but protein reagent complex has one absorbance maximum at 590 nanometer. In original old Bradford method, absorbances of standard protein solutions are measured at 590 nanometer, and the protein standard curve thus obtained is not linear, causing an incorrect protein measurement of the sample. In modified new Bradford method, absorbances of standard protein solutions are measured at two wavelengths, 450 nanometer and 590 nanometer. Then the ratios of absorbances at 590 nanometer to 450 nanometer are calculated for standards. This ratio indicates the reduction of the reagent concentration and production of the colored complex. The protein standard curve thus obtained is linear, resulting in correct protein measurement of the sample. In this slide, you see possible causes of incorrect protein measurements by Bradford method. Compare the assays times. If the total protein concentration in the samples is high, namely between 5 to 160 milligrams per milliliter, the Biret method is the best choice. If the total protein concentration in the samples is low, namely between 1 to 2000 micrograms per milliliter, then any one of the other three, namely the Lowry, the Bradford, or the BCA method, would be suitable. If the sample contains reducing agents or copper chelating reagents, the Bradford method would be the best choice. If the sample contains one or more detergents at concentrations up to 5%, the BCA method is the best choice. Each method has its own advantage and disadvantage. No one method can be considered to be ideal or the best protein assay method. The selection of a protein standard is potentially the greatest source of error in any protein assay. Of course, the best choice for a standard is a highly purified form of the predominant protein found in the samples. This is not always possible nor always necessary. If a highly purified form of the protein of interest is not available, or it is too expensive to be used as the standard, the alternative is to choose a protein that will produce a very similar color response curve with the selected protein assay method. For general protein assay work, bovine serum albumin or BSA works well as the choice for a protein standard because it is widely available in high purity and relatively inexpensive. Okay, now let's deal with our experiments. For measurement of protein concentration of samples by Bradford method, we need freshly prepared Bradford reagent, bovine serum albumin or BSA standards, and some samples. 50 milliliters of Bradford reagent is prepared by dissolving 5 milligrams Kumasi Brilliant Blue G250 dye in 2.5 milliliters 95% volume per volume ethanol, followed by addition of 5 milliliters phosphoric acid. The solution is further diluted with distilled water to 50 milliliters. A 200 micrograms per milliliter BSA stock solution is prepared, from which 10 to 200 micrograms per milliliter BSA working solutions are prepared in triplicate. Liver, lungs, and kidneys samples of Wistar rat were used. A 217 grams male healthy Wistar rat was selected. Liver, lungs, and kidneys were removed from ether-anesthetized rat by surgical operation. Samples were separately placed in phosphate buffered saline containing plastic bottles in an ice pack containing flask to keep the temperature about 4 degrees centigrade and immediately transferred to the lab. The samples were first weighed and then separately homogenized in 50 milliliters PBS by a German-made Duran pestle and mortar glass homogenizer. Alternatively, you can use a stainless steel mesh screen with a 200 mesh. The homogenates were centrifuged at 5000 G for 10 minutes. The supernatants are used for measurement of protein concentration. Please pay attention that appropriate dilution with PBS may be necessary to fall the absorbances of the samples within the linear part of the standard curve. I diluted liver supernatant 50 times due to its high protein content and those of kidneys and lungs 10 times with PBS. I used a 96 well plate for measurement of absorbances of the prepared samples. Different 96 well plates are commercially available according to the shape of the wells. A standard flat bottom, chimney well flat bottom, C bottom or flat bottom with rounded corners, V bottom or V-shaped bottom, U bottom or U-shaped bottom, and half area microplate. The first type of 96 well plate is a standard flat bottom plate. The F refers to the flat well bottom. This type of plate has excellent optical properties. It is used for precise optical measurements. It has microscopic applications for bottom observation and photography. The total volume of each well equals 382 microliters, and the working volume of each well equals 25 to 340 microliters. The next type of 96 well plate is chimney well flat bottom plate. The chimney well cell culture microplate has the same well profile as the standard F bottom plate. The difference to the standard plate is the chimney-like arrangement of the wells, namely each well stands on its own. Therefore, the risk of contamination from sample materials being carried over is minimized. The total volume of each well equals 392 microliters and the working volume of each well equals 25 to 340 microliters. The third type of 96 well plate is C-bottom plate. The letter C describes a flat bottom profile with rounded corners. The rounded corners mean that the individual wells can be pipetted without leaving a residue, and the flat bottom still enable precise optical measurements. The total volume of each well equals 346 microliters and the working volume of each well equals 20 to 300 microliters. The next type of 96 well plate is V-bottom plate. The letter V stands for the conically tapered well bottom. It is used for precise pipetting. The total volume of each well equals 234 microliters and the working volume of each well equals 42 to 200 microliters. The fifth type of 96 well plate is U-bottom plate. The letter U describes the round bottom shape. It has no sharp corners to facilitate easy and residue-free pipetting. The total volume of each well equals 323 microliters and the working volume of each well equals 42 to 280 microliters. The last type of 96 well plate is half-area microplate. For many applications, a reduction of sample volume is an important feature. Half-area microplates provide such capability. They can be pipetted automatically as well as manually without any problem and allow a reduction of sample volume up to 50%. The total volume of each well equals 199 microliters and the working volume of each well equals 15 to 175 microliters. For spectrophotometric applications, clear standard F-bottom plate is suitable. I used a biofilm polystyrene clear F-bottom 96 well plate for my work. The recommended working volume range of the F-bottom 96 well plate equals 50 to 250 microliters per well. Please pay attention that the dimensions of wells and therefore well total and working volumes of 96 well and other multi-well plates of different companies differ slightly. In each well of a 96 well plate, add 250 microliters of Bradford reagent to 5 microliters of blank, in this case distilled water, a standard or sample. I'm using a multi-channel micropipette. Okay, as you see, the color in each well changes from brown related to the blanks to blue related to standards and samples. The higher the protein concentration in the standards or samples, the more intense blue color. Place 96 well plate in the plate carrier of the microplate reader so that the well A1 is in the left rear corner of the carrier. Please pay attention to the letter A1 in the left rear corner of the carrier of the instrument. Actually, well A1 in the 96 well plate should be beside letter A1 in the left rear corner of the carrier of instrument. Don't press carrier ejection button. Instead, run the software program previously prepared for your work. For linearization of Bradford method, I prepared a software program which ran the instrument and measured the absorbances of microplate wells at 450 nanometers and 590 nanometers. For measurement of protein concentration of samples by Lowry method, we need reagent A is 2% sodium carbonate in 0.10 normal sodium hydroxide. Reagent B is 0.5% copper sulfate pentahydrate in 1% potassium sodium tartrate. Reagent C is alkaline copper solution, which is prepared by mixing reagent A with reagent B in a 50:1 volume by volume ratio. Discard after 1 day. Reagent E is falling reagent diluted in 1:1 volume by volume ratio with distilled water. Bovine serum albumin or BSA standards in triplicate. And supernatants from previous experiment. In each well of a 96 well plate, add 40 microliters blank, standard, or sample, followed by addition of 200 microliters of reagent C. Blank is distilled water. Allow the plate to stand for 10 minutes or more in room temperature. Then add 20 microliters of reagent E to each well. Allow the plate to stand for 30 minutes or more in room temperature. As you see, the color in each well changes from yellow related to blanks to blue related to standards and samples. The higher the protein concentration in the standards or samples, the more intense blue color. Place 96 well plate in the plate carrier of the microplate reader so that the well A1 is in the left rear corner of the carrier. Please pay attention to letter A1 in the left rear corner of the carrier of instrument. Actually, well A1 in the 96 well plate should be beside letter A1 in the left rear corner of the carrier of instrument. Don't press carrier ejection button. Instead, run the software program previously prepared for your work. I prepared a software program which ran the instrument and measured the absorbances of the microplate wells at 750 nanometers. For calculation of protein concentration of standards and samples by Bradford and Lowry methods, I used Microsoft Excel. Let's first see the output of the microplate reader for Bradford method. In original Bradford method, absorbances of blanks, standards, and the samples are measured at 590 nanometer. But the standard curves as obtained is not linear, and the concentrations of the samples are not correctly calculated. Therefore, Bradford method has been modified in which the standard curve is linearized. I used the latter method. In this method, absorbances of blanks, standards, and the samples are measured at two wavelengths, 450 nanometer and 590 nanometer. Then the ratios of absorbances at 590 nanometer to 450 nanometer are calculated for blanks, standards, and samples. This ratio indicates the reduction of reagent concentration and production of colored complex. First, I calculate the absorbances ratios for triplicate blanks, standards, and samples. Then I obtain their averages and standard deviations. To eliminate the background effect of blank, the average absorbance ratio of blanks should be subtracted from those of standards and samples. By this method, corrected means but not corrected the standard deviations of standards and samples obtained. To show extent of variation or dispersion of the data, corrected standard deviations of standards should also be calculated. To do so, a cross subtraction should be performed in which absorbance ratio of each blank replicate is subtracted from that of each replicate of each standard. Then corrected standard deviation of all 9 subtractions is calculated for each standard. Corrected mean of subtractions can also be calculated for each standard by averaging all 9 subtractions for each standard. Let's do it practically. Subtract the first replicate of blank absorbance ratio from the first replicate of the first standard absorbance ratio. Subtract the first replicate of blank absorbance ratio from the second replicate of the first standard absorbance ratio. Subtract the first replicate of blank absorbance ratio from the third replicate of the first standard absorbance ratio. Do the same for the second and the third replicates of blank absorbance ratio. Finally, calculate corrected mean and corrected standard deviation of subtractions. Do the same for other standards. Finally, you will obtain corrected means and corrected standard deviations of all standards. Please pay attention that the mean you obtain by this method is the same you obtain by direct averaging, but by direct method, you can't calculate corrected standard deviations. Now, draw the protein standard curve. Go to Insert, then select Scatter Plot. Right-click on Empty Chart. Select Data. Press Add. Select a name for your chart, for example, Bradford method, protein standard curve. In series X values, insert all standard concentrations except blank. In series Y values, insert all corrected means of standards absorbance ratios except blank and press OK. And again, OK. You obtain a scatter plot. For plotting best fitted curve, right-click on Markers and then select Add Trend Line. Select Linear as the software selected it by default. Select Display Equation on Chart and display R-squared value on chart. Equation of line and R-squared appear on the plot. R-squared is a parameter of correlation of data. In this case, it indicates linearity of a regression line. Press plus sign on the upper right corner of the plot and select Axis Titles. Axis Titles appear on the plot. Select names for both axes. I select name A590 to A450 for y-axis and name concentration in microgram per milliliter for x-axis. make power -1 superscript. Again, press plus sign on the upper right corner of the plot and select error bars and more options. Select direction of vertical error bars. Software selected error bars in both directions by default. I select it too. Select custom and specify value. In positive error value and negative error value spaces, separately insert all corrected standard deviations and then press OK. Now you have protein standard curve with standard deviations as error bars. You can change the appearance of the plot to satisfy your desires. Okay, let's calculate protein concentration of samples. We obtained corrected mean absorbance ratios for samples. Also, we obtained the equation of protein standard curve. In the equation, put the corrected mean absorbance ratio for each sample in place of y and then add 0.0187 to it and divide the sum by 0.0014. Because the liver supernatant was diluted 50 times and the kidneys and the lungs supernatants were diluted 10 times by PBS, multiply the respective values by 50 and 10. To calculate the total amount of protein of each sample, multiply the protein concentration of each sample by the volume of the PBS in which each sample was homogenized and divide the resulting value by 1000 to convert microgram to milligram. To calculate the amount of milligram protein per each gram of organ, divide the amount of protein of each sample in milligram by the organ mass in gram. Okay, let's calculate protein concentration of standards and samples by Lowry method. Let's first see the output of the microplate reader for Lowry method. In this method, absorbances of blanks, standards, and the samples are measured at 750 nanometer. In the next step, obtain respective averages and standard deviations. To eliminate the background effect of blank, the average absorbance of blanks should be subtracted from those of standards and the samples. By this method, corrected means but not corrected standard deviations of standards and samples obtained. Like for Bradford method, a cross subtraction is performed to obtain respective averages and standard deviations. Now draw the protein standard curve. Go to Insert, then select Scatter Plot. Right-click on Empty Chart, select Select Data, press Add. Select a name for your chart, for example, Lowry method, protein standard curve. In series X values, insert all standard concentrations except blank. In series Y values, insert all corrected means of standards absorbances except blank and press OK. and again OK. We obtain a scatter plot. For plotting best fitted curve, right-click on markers and then select Add Trend Line. Select Linear as the software selected it by default. Select Display Equation on Chart and display R² value on chart. Equation of line and R² appear on the plot. Compare R² of this line with that line obtained for Bradford method. Select names for both axes. Select Corrected Standard Deviations as error bars. You can change the appearance of the plot to satisfy your desires. Please pay attention that the standard curve of the Lowry method is not linear per se. Microsoft Excel only represents three types of lines: linear, logarithmic, and polynomial. To find the best fitted curve, it is better to use a graphical software like SigmaPlot, which is also able to give equation and R² of line. Here, for simplification of calculation, we selected linear for representing best fitted curve. Okay, let's calculate protein concentration of the samples. In the equation, put the corrected mean absorbance for each sample in place of y, and then add 0.0025 to it and divide the sum by 0.0009. Because the liver supernatant was diluted 50 times and the kidneys and the lungs supernatants were diluted 10 times, multiply the respective values by 50 and 10. To calculate the amount of protein of each sample, multiply the protein concentration of each sample by the volume of the PBS in which each sample was homogenized and divide the resulting value by 1000 to convert microgram to milligram. To calculate the amount of milligram protein per each gram of organ, divide the amount of protein of each sample in milligram by the organ mass in gram. Okay, compare the protein concentrations of samples and then protein contents of liver, kidneys, and the lungs of two metals. Please pay attention that the absorbances you measure by a microplate reader are different from those you measure by a spectrophotometer using a 1 centimeter cuvette. I remember well when I was in the lab and a young lady student came in and asked me, "Is your microplate reader calibrated?" I became surprised. I asked her, "Why are you asking me such a question?" She replied, "Because I measured the absorbances of my samples by a spectrophotometer and the results significantly were different." I told her, "Are you familiar with Beer-Lambert law and equation?" She answered, "No." I told her, "Difference between absorbances of your samples by these instruments is due to difference between light path length in 1 centimeter spectrophotometer cell and that in each well of 96 well plate. In the latter, light path length is the height of liquid in each well of the 96 well plate. Calculate the height of the liquid in each well and correct it for 1 centimeter light path length in a spectrophotometer. You can calculate the height of the liquid in each well of a 96 well plate, knowing the diameters of each well at the bottom and top and the volume of the liquid in each well. Average the bottom and the top well diameter. Find the liquid height in the wells by the following equation. Volume of cylinder equals P multiplied by R squared multiplied by H, in which volume of cylinder is volume of the liquid in each well. P equals 3.14, R is the average of top and bottom well radii, and H is the height of the liquid in each well. For more information about Beer-Lambert law and equation, please watch my previous educational video at my YouTube channel. Question: which method I used for protein measurement is more sensitive, accurate, and precise? Bradford method or Lowry method, and why? وصلى الله على محمد وسلم.